Welcome everyone. Here is another question from GATE 2024 Life Sciences. The question says, consider the following nucleotide sequence. You can go through this sequence and you will find the presence of the uracil, suggesting that this is an mRNA sequence. The other part of the question says, assume a standard initiation, standard termination, and no post-translational modification, and the average molecular mass of an amino acid to be 110 Daltons. We need to calculate the theoretical molecular mass of the polypeptide, which is going to be translated from this sequence, and we need to report our answer in Dalton. So as we all know, mature mRNA codes for a specific sets of amino acids, and these amino acids are the building block of polypeptide. We also need to understand that the translation starts at a specific point by recognizing a specific set of nucleotides, which is also called as the start codon, and generally the start codon is AUG. Similarly, the process of translation terminates at a specific set of nucleotides, which could be UAA, UAG, or UGA. So first thing, we need to figure out the presence of AUG at a 5' end, and then we need to move in a specific frame, reading frame, unless and until we encounter any of these three nucleotides. So you can go through this sequence, and what you will figure it out is there is a presence of AUG from where the process of translation will start, and then you need to move in a set of three nucleotides unless and until you encounter a stop codon. So you can quickly go through this sequence, and what we will figure it out, there is a start codon, and then you need to move in a particular reading frame, and set of these three nucleotides will code for a specific amino acid. So for example, AUG will code for methionine. Similarly, this will code for another amino acid. And in this particular reading frame, all of these sequences, a set of three nucleotides, which is also called as the codon, will code for a particular amino acid. And the presence of the stop codon will terminate the process of translation. Now, if you can look at this sequence very carefully, all of these will code for the amino acid and will be a part of the polypeptide. However, UAG is a stop codon and will not code for any amino acid. So this is the point where the process of translation will terminate. So the number of amino acids which will be encoded by this particular mature mRNA is going to be starting from here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and 14. So the total number of amino acids is going to be encoded from this mature mRNA is 14 amino acid. However, we need to calculate the molecular weight of the polypeptide, and it's given the average molecular weight of one of the amino acid is 110. And then for the 14 amino acids, the molecular weight is going to be 110 into 14, which is going to be 1540. So the theoretical molecular mass of the polypeptide translated from the above nucleotide sequence is going to be 1540 Dalton. The other thing you need to understand from this question, a very important concept, that not this entire mature mRNA is coding for the amino acids. There is a particular place from where the translation process starts and a particular point where the translation process stops. I hope this answer and concept is clear to each and every one of you. Thanks for watching the video.